Hello, my name is Tayebi. I'm a PhD graduate from Auckland Bioengineering Institute in New Zealand and a current research fellow at Matai Medical Research Institute in Gisborne, New Zealand. Today I'm going to talk about our research project, which was about integration of DTI parameters with mesh-morphing for in-depth analysis of white matter fiber tracts. Modern MR technology has evolved from qualitative, that radiologists looking at scans and report abnormalities, to quantitative methods that enabling precise measurements of tissue characteristics all around the body. This change improves the diagnostic accuracy but also introduces new challenges. There is now so much data from these scans that it's hard for scientists to handle and understand, especially in brain imaging, where lots of different types of scans are being used. Have you ever tried to solve a jigsaw puzzle with thousands of pieces? It can be very overwhelming. In modern MR studies, scientists have to work through a massive amount of data because each scan gives us a lot of biomarkers. Thankfully, there is a smart method that helps make sense of all this complex information. This technique called dimensionality reduction. Imagine being able to reduce those thousands of puzzle pieces down to just the essential ones that still let you see the full picture. That's what dimensionality reduction does with MRI data. It keeps the important information and gets rid of the rest. One of these dimensionality reduction techniques called principal component analysis, or PCA. It speeds up how quickly computers can work with scientists to pick out the most crucial parts of the data known as principal component. This is especially handy for finding issues such as brain injury or other health conditions. Diffusion MRI is one of those neuroimaging techniques that benefits from PCA. In neuroimaging application, diffusion MRI enables us to reconstruct and visualize brain fiber tracts and provide more information about the microstructure of the brain. Some of these variables used for this purpose are called fractional anisotropy, mean diffusivity, axial diffusivity, and radial diffusivity. After we take a diffusion MRI scans of the brain, scientists pick out a particular brain fiber pathway they're interested in and then apply diffusion metrics to this pathway. Currently, there are 3 different methods to get useful information from this pathway. One of the oldest methods is to calculate the average diffusion metrics as a single value over the entire length of the tract. Next method is called tract profiling, that can measure these scalar values at multiple nodes along the central portion of the tract. Last one is tract-based analysis. In this method, diffusion metric map will be projected onto a skeleton located at the center of the major fiber tracts. You can imagine these methods like a dot for single averaging, a line for tract profiling, and a sheet for tract-based analysis. However, there is a major limitation that all these methods are suffering from, and that is the loss of information due to different types of averaging method. In this study, we will introduce a 3D mesh model of the tract, which is then morphed to match the subject's geometry. You can imagine it like a 3D cube, which represents the whole volume of the tract with minimum amount of information loss. Our aim is to take the 3D model of a specific brain fiber tracts, in this case the corticospinal tract, and add detailed diffusion information and measurements to it. This process gives us a lot of data points, in this case the millions. To manage all this data, we use the principal component analysis to simplify it without losing important information. We use this technique on MRI scans of high school rugby players, and our goal is to see if we could spot any small changes in the brain that come from repeated head impacts in comparison to athletes who play non-contact sports. For this purpose, we had MRI scans of the brain of high school rugby players at preseason and postseason. We also acquired MRI scans of the brain of age and sex matched non-contact sports athletes. All scans were acquired using 3 Tesla G scanner, and multi-shell diffusion scans were acquired from all players. Briefly about the mesh-morphing method. After running automated tractography on every subject, we chose the right corticospinal tract and generated a 3D mesh model out of it. Then a CST template mesh model was used to morph to each subject's model. And after this step, both the 3D model and the MR images were aligned with each other in the same space. This allowed us to generate finite element model assigned with corresponding diffusion values. Now we're dealing with 36 CST models that are all on the same space and have the same number of nodes and elements. In order to find out about the differences between these three groups, we ran separate analysis on every diffusion matrix using PCA technique. PCA results showed us a clear separation between rugby cohort and non-contact sports athlete using the fractional anisotropy data. Similar results were found by repeating the analysis on mean diffusivity, axial diffusivity, and radial diffusivity. While separation between rugby and control group was clear, there was not a clear division between pre- and post-season scans. Additionally, using mesh-morphing technique, we were able also to visualize the distribution pattern of the diffusion matrix over the entire corticospinal tract. As can be seen in the picture, the highest change was observed along the main skeleton of the tract, which is similar to the findings from the standard tract-based analysis technique, but it provides us more details in the pattern of change. In conclusion, we introduced a novel method that intersects three scientific fields: modeling, imaging, and machine learning. We demonstrated how this method can be used to generate subject-specific model of the brain fiber tracts and subsequently extract corresponding diffusion metrics with minimal information loss. Our model's applicability was showcased by distinguishing between contact sports athletes and non-contact sports players. By applying our mesh-morphing method along with feature extraction techniques, scientists can perform predictive studies that integrates multiple quantitative variables from multimodal MR imaging. Last but not least, this study was not possible without the support of our sponsors and the Matai Medical Research Institute team. Thank you so much for your attention.